Welcome to today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Alstrom, and I'm joined today by my myeloma friends and co-hosts, including Gary Peterson, Jack Aiello, and Pat Killingsworth. So, welcome. This is the seventh in a very important series featuring the Myeloma Crowd Research Initiative. Now, if you haven't already heard about what we're doing, educated patients are connecting with top researchers to help find and fund a cure for high-risk multiple myeloma. We decided to go after high-risk myeloma because there are typically dismal outcomes for these patients with standard therapies, and it was a very attractive way to have a potentially amazing trickle-down effect that what might work in high-risk patients can also work in standard or low-risk patients. Now, that's not always the case because some targets, like we are going to hear about today, are very specific to a certain type of patient. But we're very excited about offering new solutions for these patients with very limited options. So here's what we've done so far. In February, we called for letters of intent from researchers around the world. We received back 36 very high-quality proposals. Our scientific advisory board then vetted those proposals and selected a top 10, and the ones on the shows are in this top 10. Now we are preparing to select a select number of proposals to then create fundraising campaigns around. If you'd like to watch a video that we've made about the research effort, or the MCRI as we're calling it, you can go to http and then mcri.myelomacrowd.org and watch what we have to say as co-hosts and patient advisory board members and why we're driving this research project and why we think it's important. Now, we are very privileged to have with us today Dr. Carmen Baldino and Dr. Kelvin Lee, and they are both from Jasco Pharmaceuticals and Roswell Park Cancer Institute. So welcome, doctors. Thank you for joining us.
Glad to be here.
Yep, thanks for having us.
Let me introduce you both. Dr. Baldino is scientific co-founder of Jasco Pharmaceuticals and the company's president. Prior to joining Jasco, Dr. Baldino was one of the scientific founders of China-based Biodura, an integrated R&D services company, and served as its vice president of research and business development. He also spent 10 years at Arcule, Inc., most recently as vice president of chemistry, managing a group of scientists responsible for early discovery technologies, medicinal chemistry, and analytical chemistry. He obtained his Bachelor of Science in Chemistry from Southern Connecticut State University and his PhD from Purdue and Scripps, and completed a postdoctoral fellowship at Yale. Dr. Baldino's academic research focuses on the total synthesis of natural products and bioorganic chemistry. Dr. Kelvin Lee is professor of medicine at the Roswell Park Cancer Institute, as well as vice chair of medicine and chair of immunology. His laboratory has a long-standing interest and publication record in multiple myeloma, being the first to characterize CD28's pro-survival function in myeloma. In his lab, he also studies the bone marrow microenvironment and other survival proteins, and as I just learned from him right before the show, he has been working on myeloma for 25 or more years. So,
yeah, thank you.
Thank you so much. Maybe we start with a little basic biology review, and maybe terms patients can understand, because some of this gets a little bit tricky and complicated. What typically triggers genes to become translocated in the first place? And I know this is a subset only of myeloma patients.
Mm-hmm. So let me tackle that question. This is Kelvin. Myeloma, the normal equivalent of multiple myeloma, the cancer cell, the normal equivalent is a plasma cell. Plasma cells are the cells that make the antibodies that protect you from measles, the flu, polio, etc. And plasma cells are part of a lineage of normal immune cells that are known as B lymphocytes. And it turns out that the natural biology of B lymphocytes, as they become activated, as they turn into plasma cells, is to enhance the effectiveness of the antibodies that they produce. And so they go from relatively low binding antibodies to antibodies that are much higher in affinity. But in this process of making these antibodies better, they actually splice their DNA. So they rearrange their DNA, they rearrange their chromosomes through a very controlled process that actually breaks the DNA and splices it back together again. So this works billions of times a day perfectly, but every once in a while, for some reason that we don't quite understand, this DNA splicing mechanism makes a mistake and puts a gene, instead of splicing into the right place, splices it into the wrong place. And that splicing event is known as a translocation. And it is a process that doesn't happen, the mistakes don't happen very often, but they are an outcome of really what is a normal biological process for B lymphocytes.
Okay, that's a great explanation, so thank you so much. I didn't know. I just learned that that's a pretty natural process to have the DNA break apart and join back together.
Yes.
And it makes sense that on occasion it would go wrong.
Well, it's actually surprising that it doesn't go wrong more often, given the fact that, it really is happening millions to billions of times a day. And it's surprising that the mechanisms within the body are keep this under pretty tight control.
And specifically you were studying the 1416 translocation. So have you done any? I know it's probably challenging to get a ton of samples, or even patient-reported outcomes with that translocation. But I'm wondering if you've seen any patterns and causes for that particular translocation, like a virus or a environmental or anything. Have you seen anything like that?
No, we have not. I mean, I think most of these translocations that happen in multiple myeloma are pretty random. And so there doesn't appear to be anything that really skews them in one direction or the other. Now, the interesting aspect of the 1416 translocation it is that it's fairly uncommon. 8 to 10% of myelomas actually have this translocation that causes overexpression of this molecule called MAF, which we'll talk about, I guess, in a little bit. So 8 to 10% of myeloma patients have this translocation. However, it turns out that there's about 50% of myeloma patients overexpress MAF in general, even patients that don't have the translocation. So it appears that this MAF gene is an important part of myeloma biology, even if you don't have the translocation.
And as I understood, they talked about this in the last show where we're talking about both the 1416 and the 1420 translocation. And they said for MAF can be expressed in other patients, but it's like 100-fold or significantly higher with the 1416.
Right. It's because what happens in these translocations and why they're bad or why they cause problems is they take genes that are normally in one place in the DNA and under control of the normal controlling elements for that gene. They take that gene and they stick it into another place in the DNA where that gene in the new place is way more active, and so it's being activated at a much higher level. So now all of a sudden this gene, which was in its normal place, isn't highly activated, not expressed very much. It's not being made a lot. Gets put into this other gene, which in the case of multiple myeloma is the antibody gene, and that gene is firing like crazy. And then all of a sudden, MAF, which normally isn't made a lot, once it gets dropped into abnormally spliced into the antibody gene locus, gets made way more than it should, and that's what really causes the problem.
So it just goes hyper and viral.
Exactly.
Hyperexpression.
Exactly.
Okay, perfect. Well, when the 1416 translocation occurs, do you want to talk about the CMAF and what happens there with that gene?
Well, so MAF is a gene that is falls into the category of what we call transcription factors. And transcription factors are those molecules that regulate a large panel of genes. So, for example, there are transcription factors that are specifically designed or specifically dedicated to controlling, for example, antibody production or lymphocyte activation, or a large number of genes that are required for a particular biological process. So MAF normally is involved in a lot of different biological processes in different kinds of cells. It depends on the cell what MAF is controlling. And what appears to happen in multiple myeloma is that when MAF is forced to be overexpressed by these translocations or other factors, it's thought to do three things that help the myeloma cell. One is that it makes the myeloma cell divide much faster. So now you're getting, more myeloma cells being generated compared to the normal plasma cell. The second thing it seems to do is that it allows the myeloma cell to stick better to the bone marrow microenvironment. So myelomas are very dependent on interactions with the normal bone marrow and normal elements within the bone marrow for their survival. So normally if you take a myeloma cell out of the bone marrow and you purify it away from its, all its neighborhood, they die. They don't live very well outside the bone marrow. So they have to interact with the bone marrow microenvironment. And a MAF seems to upregulate or increase the particular molecules that are involved in allowing the myeloma to stick better to its microenvironment and get more of those good pro-survival signals from the microenvironment. And the third thing it does is MAF seems to control the gene program, for lack of a better word, that is involved in invasion. So now these cells can move through the bone marrow more readily, and so they can spread farther and faster than if MAF wasn't overexpressed. So MAF seems to broadly control those three areas. Exactly how it does that is an area of great interest. A lot of people are working on that. But that seems to be what MAF is bringing to the table for the multiple myeloma cell, and that seems to be why the myeloma cells that have high expression of MAF or overexpression of MAF are more aggressive, and MAF itself is a bad prognostic factor, or that MAF translocation is a bad prognostic factor for patients with multiple myeloma.
Okay, so two questions about that. Does it then cause more? Do you see more bone disease in these patients than typical?
I think we don't the MAF population is relatively a small population, but yes, the feeling is that they have more and worse bony disease
because it's invading at a higher level.
Right.
And then have you noticed any kind of resistance to maybe bortezomib or other drugs? When we were talking in our last show.
Yeah, so this is actually what brought us in to work with Jasco. Really, my laboratory's interest is understanding why myeloma cells become resistant to chemotherapy, because for the vast majority of myeloma patients, when they're newly diagnosed, almost all of them are sensitive to chemotherapy, and most of them with modern chemotherapy, the proteasome inhibitors and the IMiDs, most of them in fact have a very good response, if not a complete response, to their initial chemotherapy. And then many of them will go on to high-dose melphalan and stem cell rescue, transplant. But the problem is that almost all patients, or many patients, will relapse. And their myeloma will come back, and when it comes back, it is more resistant to chemotherapy, although now we have other drugs to treat, and we treat that, and the myeloma will regress. But then it will come back again. So oftentimes, many of our patients have been treated with 5 or 6 different lines of chemotherapy. They've relapsed. Every time they relapse, their myeloma is tougher. It doesn't respond to chemotherapy nearly as well or nearly as long as it did beforehand. And, we have patients that have undergone 8 to 10 lines of chemotherapy, and usually by that point, their myeloma is completely chemotherapy resistant. And so really the major clinical obstacle that we are facing, or one of the major ones, is the development of chemotherapy resistance. And the question that our lab has been asking is, how does that happen? What are the molecules that are involved that make these myeloma cells more resistant? And just a little bit of background on how we ended up interacting with Jasco is that one of the molecules that we study is a receptor on the surface of myeloma cells called CD28. Now, CD28, back in when I first started in science, really was mostly and has been mostly described as a pro-survival for T lymphocytes, and it's very important for T lymphocyte activation. But what we've described since 2007 is that CD28 is a critical pro-survival receptor for multiple myeloma. And as myelomas get treated with chemotherapy, more and more of them become CD28 positive. And so when you're newly diagnosed, about 25% of cases have high expression of CD28. By the time you've had your first relapse, about 50% are CD28 positive. About your second relapse, or as you relapse outside of the bone marrow, it's about 80 to 90%. All of the very late-stage myelomas, the secondary plasma cell leukemias, they are all CD28 positive. And other groups in Spain and France have shown that CD28 expression is a poor prognostic factor for multiple myeloma. And what we've discovered in the lab is that when CD28 gets activated by interacting with the normal bone marrow microenvironment, it transduces a pro-survival signal that makes the myeloma cells completely resistant to everything we've actually tested them against, including bortezomib, dexamethasone, the steroid, arsenic trioxide, melphalan, which is commonly used or characteristically used in the transplant setting. So all the chemotherapies that we've tested it on were that CD28 confers significant resistance to those chemotherapies, except for the PIM2 inhibitor that Jasco generated. So the JP11646, we were initially interested in it because Carmen and Jasco identified it being overexpressed or effective against MAF translocated myeloma cell lines. Now, we also know that CD28 is significantly overexpressed in the MAF translocated subset, or the MAF gene signature subset of myeloma. So there was a connection between CD28 and MAF. And so we tested Jasco's PIM2 inhibitor on, to see if it could overcome CD28-mediated protection against chemotherapy, and it was the only drug that could do it, which was striking to us, and really from a scientific and research standpoint, really, it really compelled us to go forward and take a much better, a closer look at what this PIM2 inhibitor was doing.
Can you give us a little background about what the PIM kinase inhibitors do and how they work?
So, Kelvin, why don't I start to answer this question? Mm-hmm. This is Carmen. As you can see, we found a great collaborator in Kelvin to work with on this program.
Yes, you did.
So let me start with a brief description of the PIM family of serine threonine kinases. There are three isoforms, PIM1, 2, and 3. There's a high degree of homology amongst the isoforms and a redundancy in the roles. Interestingly, the PIMs are also constitutively active, which means that if they're present in a cell, they're active and operating within that cell. The PIM kinases are involved in regulating several signaling pathways that are fundamental to cancer cell development and progression. The PIMs have also been shown to be overexpressed in multiple cancer types, including liver, pancreatic, and hematologic malignancies, especially multiple myeloma. In fact, it's been reported in the literature that PIM2 activity is required for survival of multiple myeloma cells. So the PIM kinase inhibitors to date have all been small molecules, as JP11646 is a small molecule, and these inhibitors bind to the PIM kinases and inhibit the normal function of the protein. Our drug, 646, has proven to be unique when compared to other PIM inhibitors described in the literature. Our drug is the only inhibitor that is able to inhibit the PIM's action in an ATP non-competitive fashion. What that means is that in a cellular environment, JP11646 will not be in competition with ATP for binding to the PIMs. And this gives it a distinct advantage when compared to other inhibitors. Secondly, our drug has not only demonstrated an enhanced ability to inhibit the PIM signaling pathways, but also the ability to nearly eliminate PIM expression levels in multiple myeloma cells. This is in contrast to other PIM inhibitors that have been shown to actually significantly increase PIM expression levels in multiple myeloma cells. We've confirmed this in a number of multiple myeloma cell lines in collaboration with Kelvin's group, and consistently our drug has shown to dramatically decrease the PIM expression levels, while the other PIM inhibitors increase and in certain cases more than double the expression level of the PIM kinases. And Kelvin, I know that you continue to work and better understanding the mechanism, and maybe you can provide a little additional information on some of those findings.
Sure. I mean, so the mechanism of action of, as Carmen alluded to, the mechanism of action of JP11646 seems to be very different than the other PIM2 inhibitors that are currently under development by other companies. So one of the things that we expect it to do, which it does, both in cell lines and also when we've tested it in animal models, is that it blocks the activity of the kinase activity of the enzyme. So it is the traditional or characteristic enzyme inhibitor. The one thing that it does that is very different is that through mechanisms that we are still trying to work out, it shuts off the expression of PIM2 at the gene level and at the protein level. So not only does it block the activity of the enzymatic activity of the molecule, of the protein molecule, but it turns it off. And we are working on, and that whole area of how, as Carmen mentioned, PIM2 is normally, if you make it, it's on. So what controls the activity of PIM2 is how much is there. So if you can turn it off and block its activity of whatever else is there, then you have a sort of a doubly effective mechanism. What is controlling PIM2 expression is not very well known in the entire PIM2 field. Well, it's something that we're trying to work out, but this seems to be a very different way of reducing PIM2 activity compared to the other PIM inhibitors.
Okay. And just a basic question on kinases. Kinases are related to the signaling pathway. They are the signaling pathway. What exactly are they?
They are the signaling pathway. So a lot of how cells signal internally, we tend to think of it as wiring, but it's not really wiring. It's really very typically it's a enzyme that can add a phosphate group onto another molecule, and by sticking that phosphate atom onto another molecule, it changes the quality of that molecule, and that molecule then usually is an enzyme itself, which now gets activated by the fact that the kinase stuck a phosphate on it, and it now goes and activates its target, and that's how the signal sort of moves from the surface of the cell all the way into the nucleus where the DNA and gene regulation is taking place.
Okay, and it sounds like it's just shutting it down from what you were saying earlier. It's shutting it down at an earlier stage in the process, so it's affecting the gene and the protein. So you're kind of like trying to stop it at the source. Is that a correct way of saying it?
It's a double whammy against PIM2.
Okay, that makes sense. So how did the two of you get connected?
Well, I think, Jasco, this is Carmen again, and we founded the company with really an expertise in medicinal chemistry and early drug discovery, but our mission was really to address unmet patient needs. And as a small company, we weren't going to be able to build all the expertise internally, and so we identified the approach of going after a strategic alliance to bring in that clinical expertise and a knowledge of the unmet patient needs. And as I think as Kelvin sort of alluded to, we identified Kelvin's research as interesting and well connected with what we were doing. And I think from our very first meeting up in Buffalo, we found that there was just a perfect match of not only scientific interest, but also a desire to bring new approaches, new treatment options for high-risk multiple myeloma patients.
Okay, great. And Dr. Baldino, how did you develop this compound in the first place?
So the compound was developed at Jasco. As I said, the company was founded with an expertise in medicinal chemistry and also in the design and optimization of enzyme inhibitors. So the first thing we did at Jasco is to build a proprietary library of small molecules that were fashioned with binding elements in order to bind to kinases. We then took a small subset of those compounds and screened them against a panel of oncology-relevant kinases and identified a number of areas where we were inhibiting the kinases. Again, as a small company, we had to be very careful about the kinase we chose to work in. So we were inhibiting some kinases such as EGFR, CMET, PI3K that were in very crowded areas. A lot of larger companies involved in that. The initial PIM hits were micromolar inhibitors of the PIMs, but the PIM kinases were fairly novel. And there was some clinical validation in the literature. But I think more importantly, we had identified that PIM2 was implicated in multiple myeloma, and so we felt that this was a good match of the science and our mission as a company. So we took those initial hits and went through a lead optimization phase, and were able to optimize the potency of the compound against PIM2 from a single-digit micromolar inhibitor down to a 500 picomolar inhibitor, and actually drove the activity in multiple myeloma cells down to 12 nanomolar. So we had a very strong optimization program. And again, this is sort of the point where we met with Roswell and talked things over with Kelvin's group and began to work together to now move these effects into more sophisticated animal models and really focus on how to develop the program for high-risk multiple myeloma patients. Kelvin, maybe if you want to say a few words, I know you've already mentioned the CD28 assay,
Sure.
But a few words about our initial interactions.
Sure. No, it's working with Carmen and Justin and Jasco has been a real treat. So as I mentioned, we initially were very interested in what can overcome what we think is a dominant mechanism of chemotherapy resistance in multiple myeloma. So we looked at the JP11646 ability to overcome CD28 mediated resistance, and that was very exciting. Then we subsequently have done a number of studies also with the Center for Drug Development here at Roswell Park, looking at both the mechanism of action, how does it work, and how is it inhibiting the survival signals that are getting into the multiple myeloma, which I talked about a little bit, and also how effective what is it in animal models of multiple myeloma, and we've done a lot of those studies trying to determine the right dose and schedule and toxicity that's involved. And we found, at least in mouse models of multiple myeloma, that at a 15 milligram per kilogram dose, which is reasonably well tolerated by the mice, give it twice a week, Mondays and Tuesdays. Essentially, about 60% of the mice were being cured of their myeloma, and these were not little myeloma tumors. These are pretty big myeloma tumors that they were being cured of. So, at least in mouse models, the drug appeared to be very effective and with pretty well tolerated side effects. Now, we also found that it could be combined in those mouse models. It could be combined at a lower dose with dexamethasone, which is commonly used in the treatment of multiple myeloma. So that combination also seemed to be very well, even better tolerated, and have the same or a similar degree of effectiveness against the multiple myeloma.
Okay, well, let me back up and ask a question about chemo resistance overall, because we've seen this as a recurring theme, I guess. There are a lot of people who want to overcome chemo resistance, find out what cells are surviving all the chemo, and then study those. So is the goal to enable the chemo to work and then get rid of all the residual myeloma cells, or in other words, do you bring a high-risk patient back to normal or low risk? Or are you looking for how do we cure this patient, forever?
I think it's actually both. So I think that, and the reason, and that's a great question, I think that there are, in fact, two mechanisms that are at play. One is that it's very clear that myeloma, as you know, is really not one cancer, but is really a whole tribe of different kinds of cancer cells, and that's what the genetics and the gene profiling research has shown us. And it's really a tribe of different myeloma cells that have different resistance to chemotherapy. So as you beat on the myeloma with chemotherapy, the ones that are resistant to the chemotherapy now can continue to grow, where the ones that are sensitive get killed off. So one strategy, which we are pursuing, is can you identify the molecular mechanisms by which those resistant myeloma cells are being resistant? What's their basis for the resistance? And can you target those molecular mechanisms to now to do exactly as you were saying? Can you now resensitize all those myeloma cells to chemotherapy that they were previously resistant to? So can you reset the clock back to when they were newly diagnosed, for example? So that's one strategy. The other strategy is to actually find compounds like this PIM2 inhibitor, which we think bypasses all those resistance mechanisms and will kill cells regardless. So that's a different mechanism, and that's finding a drug that's not affected by these pro-survival mechanisms, chemotherapy-resistant mechanisms in multiple myeloma. So I think that those are two approaches. They are linked. They're not exactly, they're conceptually a little bit different, but I think that both of them, in fact, are actually aiming to cure patients, and I think that chemotherapy resistance is, in fact, probably one of the major signatures of high-risk myeloma. It's really those myelomas that can adapt very quickly to the chemotherapy that they're being faced with that cause us the major problems.
And, well, I want to go back to what you said earlier, because you said CD28 expression goes up and up and up while it's present in this 14-16 translocation. You keep seeing increases as people keep relapsing. So this may not just be some solution for 14-16. It might be a solution for everyone. Is that correct?
Exactly, and that's why the additional excitement, because we don't think that this drug, the PIM2 inhibitor is limited to 14/16 translocations. We actually think that it is more broadly applicable because CD28 is expressed on lots of different kinds of myelomas. And also, again, I alluded earlier that MAF overexpression seems to be detectable in 50% of myelomas, even though the translocation is seen at a lot lower level. So the fact that this drug might affect MAF-related biology, in addition to anything that is associated with the translocation, means that there's actually a fair number of myeloma patients that this drug might be very active in.
Yeah, that'd be great. That'd be terrific. So it sounds like you've run mouse models. Can you kind of walk us through where you're at in your process?
So right now we're in preclinical. This is Carmen. We have a very comprehensive package of data, from PK studies and efficacy studies, mechanistic studies continue to move forward, and we're ready to move into our GLP tox studies, which is really the last remaining study before we're able to file an IND. We have a very good plan on how to move forward in filing from where we are today to filing the IND. We believe it to be about a 9-month process. We have very strong interest from the clinical community and a number of different clinical sites and investigators willing and interested to participate in the trials. Of course, we view Kelvin as a key driver in that process and Roswell Park. Yeah, as a key component to that process. But we believe we're 9 months away from filing an IND and moving forward. I don't know, Kelvin, if you want to add anything about the interest level from your side.
It's extremely high. I mean, again, like I said, the patients that I treat that are that have the biggest clinical challenge, and the biggest challenge are those patients that have chemotherapy-resistant multiple myeloma, and those are the patients that we really need good options for, and that we really need new approaches for. So we're real excited to do this trial. It's one of those. I suspect we could have this trial for phase 1, completely accrued within 3 months. I mean, you're probably shorter than that if depending on the trial design. But we have a lot of patients that this PIM2 inhibitor, has a tremendous amount of potential for.
Okay, that's amazing. So the IND. What does that stand for? And then how long between the time you file for an IND can you get a clinical trial started? Because that's a process in itself, right?
Well, IND stands for Investigational New Drug Application. That's filed with the FDA, and the way it works, if they don't contact you within 30 days of filing that document, that means you're okay and ready to move into phase 1 trials. So I think a lot of the legwork it comes in putting the IND package together, which will have a description of the clinical protocol and all the information that you've compiled during your preclinical work. So it really is kind of a 30-day waiting period. As long as everything passes through the FDA without a hitch, you're able to move forward into your clinical trials. And usually there's an intermediate step where you have an interaction with the FDA as you're preparing the IND, so that some of the questions can be answered, and at the point where you file the IND, things go fairly smoothly.
Mm-hmm. And of course you're working with mice, so they might not be telling you about their side effects. And this isn't in phase 1 clinical trials yet, but do you have any indication about what potential side effects could exist?
Yeah, so at this point, we've done a number of initial toxicity studies in both rodents and dog, and what we've demonstrated is that doses that provide efficacious plasma levels of JP11646 are well tolerated by the animals. Of course, that's a good first step, but we need to move into the GLP tox studies. We have protocols for those studies that have already been designed. The studies take about 6 months to complete, and then the results from those studies will really give the answers to this question and help us to design the clinical protocol as we move forward into those trials. And then really the key question is how is it tolerated by humans as you move into the phase 1 studies.
Right. And one interesting observation that we've made is that we've actually looked at the effect of JP11646 in normal human blood mononuclear cells, so the white cells in your blood. And those cells, in fact, express PIM2, but for some reason, JP11646 doesn't shut off PIM2 expression in normal cells, or at least in normal blood cells. So there appears to be a differential mechanism where it turns off PIM2 expression in myeloma cells, but doesn't seem to do the same thing in normal blood cells. So that is promising to us that the side effects will be, reasonably modest with this drug.
Well, that'd be great, because we've heard about some other drugs that could potentially, depending on what target you go after, could potentially be damaging, and we don't really want that.
Right, exactly.
So I think just a couple more questions from me, and then I'm kind of hogging all the time. So would this work for the 1420 MAF-B translocation as well, or would it mostly be present in the C-MAF, which is the 1416?
I don't think it's going to be, because as you alluded to earlier, I think it has efficacy that is beyond just MAF, right. And so my guess is that it will work in all the MAF translocations, because I don't think it's working on MAF specifically. I'm thinking it's working on what MAF is doing to the cell, and so as long as the MAFs are going through a common pathway, and if other processes like CD28 is going through that pathway, JP11646 is going to be able to target that. So I think that it's not just CMAF that is going to be targeted by this drug. I think other processes that are important for the survival of the myeloma are going to be targeted too.
Mm-hmm. Perfect.
And I think we've also shown, just to add, we've also looked at a panel of multiple myeloma cells and showed kind of broad efficacy. We've had good efficacy with cells that have the T414 and the T1114 translocation. Unfortunately, we haven't tested a cell line with the T1420 translocation.
Okay. Well, great. Jack, would you like to ask any questions that you have? And we'll go through Jack, Pat, and Gary and see if they have other questions.
Sure, Jenny, I'm happy to, and thank you very much for letting me participate in this. Some of the questions I had have already been asked, so I wanted to ask some questions related to better understanding the biology, I guess. Can you explain to me what's the relationship, if there is one, among MAF, PIM2, and CD28?
That's a great question that we don't know, because that's what we are trying to figure out. So the literature currently, the literature does not connect. There's no scientific research that says that PIM2 and CD28 are connected. Although our data suggests that those two are connected, that PIM2 may be somewhere in the CD28 signaling pathway. And there's very little literature between how MAF and CD28 is connected, although, again, our data suggests that MAF is regulating CD28 expression or activity, and there's not very much data connecting the MAF and PIM2. But all the sort of work that we've done suggests that they all are in the same bucket. They are all that there are interactions there that simply haven't been identified yet, but they are all interacting with each other. One of the things that seems true is they all travel together. They travel in packs. So if one looks at, say, the myeloma cells from patients that seem to have overexpression of MAF. You also see that those cells overexpress CD28, and those cells have overexpression of PIM2. So they tend to travel in packs, which says to me that they are all related to each other, and they're connected to each other. We just don't know what that connections are yet.
Thank you. In the letter of intent, you wrote that the treatment of cells with JP11646 at low NM levels rapidly decreases PIM2 protein. Low NM levels, is that the micromolar? Is that just basically the dosage level of the JP11646?
Yes, that's nanomolar. At nanomolar levels,
okay, right.
When cells are treated with the drug, it decreases the PIM expression levels.
And then you followed that by saying, interestingly, this happens in cells but not PMBCs. Is that the same response you were explaining to Jenny earlier about?
Okay. PM, or that stands for peripheral blood mononuclear cells. So those are human white blood cells, essentially.
Got it. And currently, is Jasco Pharmaceuticals funding all this research?
So yes, we have funded all of the research to date, and we continue and are dedicated to moving this program forward. We're thrilled to be working with Roswell Park, and we, like every small company, are looking for multiple mechanisms to be able to move the program forward.
Thanks very much. I'll let someone else ask questions.
Oh, Gary, you had several questions.
Yes, I do, and thank you very much. My lawn guys are out there, so hopefully I don't have any background noise, but I'll kind of go from there. In some of the literature, and there's not a whole heck of a lot, I guess, because you're relatively new. You can Google JP11646, and there's just a handful of information. But online, it has shown activity against solid tumors, including breast, colon, liver, lung, and pancreatic cancers, as well as multiple myeloma. Mm-hmm. So what have been the results for these other cancers, or has there been, and have some gotten to human trials?
Yeah, so this is
with
Carmen.
JP11646, does it have efficacy for other myeloma fish abnormalities other than 1416, or could it be beneficial for low-risk disease as well?
So let me start to address the question. That's a good question. So in our work with Roswell Park, there was an interest to study the compound in other cancer types. In all the ones that you had mentioned there, it's known that PIM2 is overexpressed, or the PIMs are overexpressed in those cancer types. So again, we were able to confirm that in a cancer type where there's overexpression of the PIMs, the drug JP11646 has efficacy. So all of those studies are sort of at a preliminary stage. We as a company focused on multiple myeloma with the potential to show an enhanced effect within this high-risk MAF subgroup. So kind of our mission was to identify a critical need and focus our effort in that area. And as Kelvin, and maybe Kelvin you can add to this, but as you've already mentioned, that we've also demonstrated broad efficacy within the different multiple myeloma subtypes. So we think there is applicability broadly for multiple myeloma and the potential in other cancers, but our focus as a small company is to drive the program as quickly as possible to the clinic for these high-risk patients.
Yeah, I think that it will, as Carmen alluded to, I think it will have efficacy, or does have efficacy against both other translocations, so both high risk and low risk. And our initial testing suggests that it is at least, 10 to 100-fold more potent than the other PIM2 inhibitors that we've tested that are also currently out in clinical testing. Now, most of the other pharmaceutical companies had focused on acute myeloid leukemia and their PIM2 inhibitor program. So there's not very much going on in multiple myeloma, although there is suggestion that other pharmaceutical companies are also interested in that. AML seems to be another target, and certainly, as Carmen alluded to, we see broad activity in a wide range of solid tumors, lung, prostate cancer, bladder. So it is a drug that, in addition to its efficacy in multiple myeloma, may be broadly effective in a whole range of cancers.
Could wipe cancer clean, huh?
Well, I don't know about that. Although that
somebody
means
walks in, they've got breast, colon, liver, lung, pancreatic, and multiple myeloma. Boom.
Give them one dose.
One size fits all.
Yeah, that way I could retire and just go drink margaritas on the beach or something. That'd be great from my standpoint.
Or sell your company to Google for $400 billion.
Yeah, well, I'll let Carmen worry about that part.
Yeah. No, I think
well,
what we
one other
try
question,
to do,
and this is, and this could be a stupid question, but I hear so many times that drugs are tested in vitro. And then somebody says, but unfortunately, whenever you put the drug into a human, the bone marrow microenvironment creates an issue that you can't duplicate in vitro. And as a result, I'm thinking to myself, well, then why don't you throw a little bone marrow in that darn test tube?
Well, that's a great question, and that's actually one of the big challenges for the multiple myeloma field is that we don't have yet, although people are developing great models of how the myeloma interacts with the bone marrow microenvironment. That having been said, we believe, and we're probably biased, that CD28 is one of those major interactions that happens between multiple myeloma and the bone marrow. And so if you can target anything that CD28's doing, you're essentially targeting how the myeloma interacts with the bone marrow microenvironment. So we think that that's one of the reasons why we think that this PIM2 inhibitor has real promise, because it goes after one of the key communication pathways between the myeloma and the bone marrow microenvironment.
Okay, thank you. One last question, and maybe a short follow-up. You want to study the role of the PIM kinase and how they play in CD28 signaling pathways. Mm-hmm. And that's a survival mechanism that is a major cause of resistance frontline multiple myeloma treatments. And what percentage of myeloma patients have this CD28 signaling pathway? I think you said something on the order of, depending on how many times you've been treated, it becomes more and more.
Right.
So likelihood is that this becomes more and more important the later you are in your stage of myeloma development?
Exactly right. So it starts off, about 20%, but as people relapse, it essentially, by the very end, really almost 100% of patients multiple myeloma CD28 positive. So again, we think that anything that can interfere with that pathway, like JP11646, is going to be very useful with that later stage chemotherapy-resistant multiple myeloma.
Okay. Well, that's very, very interesting. And, I'll leave it up to Pat because I see we're running out of time.
Pat, did you have some questions?
Well, my first thought, Gary sort of kidded about this with the Google thing, was, so why isn't Novartis or Celgene backing up with a dump truck full of $100 bills? This sounds too good to be true. So what are some of the possible sticking points, some of the landmines?
Well, I'll just make one comment, and then Carmen knows this field better. My impression has been that large pharmaceutical companies are risk-averse, which is why what you guys do is so important. Large pharmaceutical companies want to see the phase 1, phase 2 study done and then dive in. And they don't want to assume the risk of very early stage development. So that's where the big gap is, and this is why I think you guys are doing phenomenal work, because this is the hole where, Celgene's not, or, Amgen or, Bristol Myers Squibb's not going to come in and say, hey, you have a product. We don't know if it's going to, be, how it's going to work in patients, but here's $bazillion just on promise. Right, so that's just not the way that pharmaceutical companies who have to make money, which is what their shareholders want. That's not how they operate. So they don't really like taking risks on early development. Again, which is why I think what you guys do is phenomenal.
So let me add to that answer, and I think that Kelvin is right that a lot of companies have talked to us, and the PIM area is becoming very exciting within the pharmaceutical and biotech industry. So there are a lot of folks getting involved in the area, and there is interest. And I think what a lot of companies and groups would like to see a program move a bit further, get at least to the IND stage, start some clinical trials efforts before they jump in. That can always change, but I think the challenge for a lot of small companies like Jasco is to find a way to continue to progress the program until somebody makes the decision to jump in and help out. So I think ultimately to bring a drug to market, a small company like Jasco is going to need help, and we continue to pursue that help. But it's sort of a juggling act where you're trying to find ways and continue to progress the program to answer questions and to reduce the risk, and so that people are able to then jump in and help out. So we're very positive about the program. We are dedicated to finding those mechanisms, and we continue to have people like Kelvin who give us a lot of excitement about moving the program forward, and that there's the potential that it can do some good. So that's what drives us. But it's kind of the way the industry works. That small companies have to prove things in order to make that program move forward, and that's what we're doing.
Sure, I get it, but this sounds pretty darn good. So let's say that you get some help from people like ourselves and maybe others, and things start progressing. As a high-risk, late-stage myeloma patient myself, so how long until something like this might actually, I'm not sure FDA approved, but at least get to a point where there would be enough clinical trials that it could be helping patients?
So, as I mentioned, we believe we can complete the preclinical and file the IND within 9 months. A phase 1 trial for high-risk multiple myeloma patients could initiate a month after filing that IND, and that phase 1 study, on average in the industry would take about 12 months. Could be shorter if you're optimistic. And then, you can move into a phase 2A study where you're treating a larger group of patients. So, within a couple of years, you're going to be in. You have the potential to be in a larger group of patients.
Sure. So 3 or 4 years, and this thing could really be rocking and rolling.
That would be great.
I wish you guys the best of luck. Thanks for including me, Jenny.
Oh, thank you. And I just want to comment. I think you're in the classic. What's called the Valley of Death. So I interviewed Craig Cruz about carfilzomib, and that stage where you first start testing compounds, you have that early pharmaceutical company that's working and growing, and then you start showing results, and you move to different sources of funding. There's this classic gap that exists, and so it sounds like you're in that gap that needs to be filled to be able to move forward.
Yes, exactly. And again, like I said, this is exactly what we think that your guys' efforts is phenomenal.
Well, if no one else has any other questions, I'll ask one last question. You talked a little bit about this. Can you walk us through just your next step milestones, and then just an estimated budget of how much you would need to get those steps finished or completed?
Sure. Happy to do that. So really the next key milestone is to complete the GLP tox studies, and then to use that data to compile our IND package and file that. As I've said, we think that's about a 9-month process, and we believe we can get that done with about $800,000. The next milestone would be to then initiate a phase 1 study for relapsed refractory multiple myeloma patients. We would look to enhance the patient population to have enough of the MAF subgroup patients in order to show an enhanced effect in that subgroup. And using sort of industry averages, you'd look at about a 12-month study there, and you'd need about $2.1 million to complete that study.
And do you usually have pharma partners at that point, or you would still just drive that yourself?
We're as a small company would be happy to have a pharmaceutical partner at any point along that path. As we gained a lot of knowledge working with Roswell Park and Kelvin's group, we'd be thrilled to have another group come in with more expertise to help guide and move the program forward. Our goal is really to find the best path forward for the program, and certainly would be happy to have a partner to help move that along.
I'm just curious about how that works. Well, Dr. Baldino and Dr. Lee, we are so thrilled that you're joining us today. This is very exciting stuff. We're very excited with what you're doing, and hope your work will continue, and hope we can help you.
All right, great. Thank you very much.
Yep. Thank you. Enjoyed the discussion. Yep.
Okay, thank you so much for listening to the Myeloma Crowd Radio and the new MCRI series. We know that patients can help support the discovery of a cure, and we encourage you to become involved.
Lucky Land Casino, asking people, what's the weirdest place you've gotten lucky?
Lucky? In line at the deli, I guess?
Aha, in my dentist's office. More than once, actually.
Do I have to say?
Yes, you do.
In the car before my kids' PTA meeting.
Really?
Yes.
Excuse me, what's the weirdest place you've gotten lucky?
I never win and tell.
Well, there you have it. You can get lucky anywhere, playing at LuckyLandSlots.com. Play for free right now. Are you feeling lucky? No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.